So let's take a look at that. So off we are to semi-conservative replication. What we realize immediately is that simply unzipping a DNA strand is not good enough. It would take too long. It would take years to replicate all of the DNA in your cells. So what we have found instead is that DNA has evolved this or DNA polymerase has evolved this very clever structure of making what we call replication bubbles. And in essence, what happens is you get the beginning of two forks, one here, proceeding in opposite directions to each other, so that your DNA can form multiple replication bubbles and therefore form multiple replication sites. And so your DNA can open twice as fast in many different places. So we have now we know the structure of DNA. Now we have the formation of replication forks. Let's take a look at how that happens. Well, within the replication bubble, DNA unzips, if you will, first uncoils by the action of an enzyme called helicase. The helicase opens the DNA up, and the replication forks begin. We have what is called an origin of replication. Now, here's the thing. Remember 3 rules about DNA replication and DNA polymerase. Number 1, polymerase reads the strand in the 3' direction to the 5' direction, which means that it adds just the opposite of that. It'll add 5 to 3. So as it polymerizes and puts those babies on there, it can only polymerize with a 5 to a 3, which means that it needs that 3, look at that DNA molecule, that nucleotide molecule, it needs that OH group hanging off there to polymerize in that direction. So it adds 5 to 3, reads 3 to 5. Okay. And it needs that 3' OH group hanging off to add that next phosphate group onto. All right, well, that being said, well, let's stay here. We therefore have a problem at the replication fork. Forget this side for a second and let's look this way. We have 2 strands. We're going to call one the leading strand, which is the one that DNA polymerase can read smoothly, and we have the lagging strand, the one it cannot read smoothly. This one, obviously, it's going to read 3 to 5 and add 5 to 3. This one, since it's the anti-parallel of the top one, reads in the opposite direction. Tough stuff for DNA polymerase, but there is an answer. First, though, how does it all begin? It begins right here by the adding of an RNA primer. We have to add a piece of RNA there, our primer, by the action of primase. Or the RNA primer is added there, and now DNA can begin polymerizing. Because think about it, there was nothing to polymerize to, so the RNA primer is there. Okay, now DNA in the leading strand DNA polymerase in the leading strand can easily read down that. That would be the end of the story, except the DNA polymerase is going like this, and in going like this along the replication fork, it can't read this bottom strand, but it does. And the way it does is by the formation of Okazaki fragments. Okazaki fragments, small portions of DNA, that each of which are going to be primed separately by RNA primers. So therefore, we're going to put an RNA primer here, and add a small group of DNA nucleotides at replication fork, which of course was done when the replication fork was further, if you will, upstream, and further, if you will, upstream. And so we have a series of RNA-primed Okazaki fragments that have to be stuck together. Easy. Along can come DNA polymerase and remove those RNA primers, and then along can come ligase and glue those Okazaki fragments, glue, little quotes there, together, making this one smooth, continuous strand. So what do we have? When we're at the replication fork, we have 2 things happening. Number 1, we have the DNA polymerase moving down, reading 3' to 5' very smoothly. We have the formation of Okazaki fragments. The Okazaki fragments are first edited, the RNA primer is taken off, and then ligated, put together. When we are done, ta-da, we have 2 smooth strands of DNA template that's been added. What's the rest of the story? The rest of the story is editing. Remember, there are enzymes that can do mismatch repair, and that's the DNA polymerase, and excision of any mistakes. And last but not least, what happens when we come to the end of our DNA strand? Well, what happens when we come to the end of our DNA strand was a real problem. And that problem was this. Since it's the end of the DNA strand, you're going to run out of, and the beginning, you're going to run out of places to hook to polymerize to. And so you lose chunks. But chromosomes and DNA is no dummy. What they can do is they have a series of repeated units, and these repeated units are called telomeres. And telomeres are little safety things, so to speak, so that even though you're going to lose a little of DNA, it won't be coding pieces of DNA. And therefore, we have end safety mechanisms called telomeres. Well, in a nutshell, and we could tell you a lot more, in a nutshell, that's the way DNA replicates. Now we're going to have to think about one more thing. If that's the way it replicates, how does it work? We'll take a look at that later.